Okay, this is a second video about the digestive system, talking here about the enzymes involved in digesting each of the types of macromolecule. At the beginning of the trip in through the digestive system, we're ingesting our food in the mouth. The first thing we do is chew, and the salivary glands, which are accessory organs, accessory glands, produce saliva, and saliva helps to lubricate our food, but also contains an enzyme, salivary amylase. Salivary amylase is an enzyme that works to help hydrolyze or digest or break apart carbohydrates. Salivary amylase can do its job as the food is going down the esophagus and into the stomach, but in the stomach it will be digested by an enzyme that's in the stomach called, it's a protein digesting enzyme, so I'll get the right color for that. The protein that's digesting enzyme that's in the stomach is called pepsin. Pepsin is produced in a form that is not active, pepsinogen. Pepsinogen is produced throughout the day and turns into pepsin in the presence of acid. So when the pH is low. The acid in particular is going to be hydrochloric acid because that's what your stomach makes, but any acid will do the job. So when the pH is low, when the pH is very acidic, then pepsinogen will turn into pepsin, start digesting proteins, start taking polypeptides and breaking them into smaller polypeptides. We continue through the stomach. At this point, we haven't started the chemical digestion of lipids or nucleic acids yet. When the stomach is done its job, and we know that because the pH will drop, the stomach will empty, go past the pyloric sphincter into the duodenum. As the stomach empties, that also sends a signal to your gallbladder and your pancreas to start secreting their various substances. The gallbladder secretes bile. Bile, remember, is there to emulsify lipids, to break them into small pieces so that the enzyme can reach them, so that they're not, if they're looking at the center of a big blob of oil and the enzyme's trying to get to it, it won't be able to. But if you have many small droplets, it's much more efficient digestion. So the bile will be squeezed out of the gallbladder, go down into the duodenum. Similarly, the pancreatic juice, which is everything made by the pancreas, is going to leave the pancreas, go through the same duct into the duodenum. The pancreas secretes digestive enzymes that work for all of the 4 types of macromolecules. It secretes some carbohydrases, and this is pretty vague. It secretes pancreatic amylase, which replaces the salivary amylase that has been damaged in your stomach. These ones here come from the pancreas, are activated in the small intestine. Your lipid digesting enzyme that's made by your pancreas is called, it's either called pancreatic lipase or just lipase. In this course, we can just call it lipase. So this also comes from the pancreas. Nucleic acids, we're just going to say there are a bunch of them, but we're going to say the enzyme involved is called a nuclease. And there's more than one of them because DNA and RNA are different, so we'll call them nucleases, and we'll leave it at that. Just one word to remember. For proteins, we've got more to remember. We've got pepsin already made in the stomach, but from the pancreas and the small intestine, we have other ones like trypsin. We have chymotrypsin. We have erepsin. And we have various, we can call them proteases, which are enzymes that digest protein, or we can call them peptidases, which are enzymes that digest peptide bonds, that break peptide bonds, that hydrolyze peptide bonds. So you can see that for proteins, we have a much larger list of enzymes required. One thing to remember about these enzymes is what they work on. So here we have them organized into the 4 different types of macromolecule, but also where they're produced and which pH they work best at. That seems like a lot of information, but the pH is not much extra information at all. Because in the mouth, this region is pretty close to neutral, so around 7. pH somewhere around 7. As we go down the esophagus, that stays about the same. As we go into the stomach, this is the part where we have a pH that's low, a pH of around 1 or 2. This is the only place where things are that acidic. But as soon as we come into the duodenum, the pancreas secretes not only its pancreatic juice with enzymes in it, but also this ion, the bicarbonate ion. So bicarbonate is a buffer. And buffering, when we're talking about acids and bases, is bringing towards a central pH, whether you're looking at an acid or a base. And so bicarbonate will buffer the pH to around 8. So we can expect that the pH in the mouth is about 7. And then 7, 7, 7, 7, 7, 1 or 2 in here. In the stomach, somewhere between 1 or 2, very acidic. And then as soon as we get into here, a pH of around 8, and that will continue the whole way through until the end. pH of around 7 or 8 at the end. So if you remember where the enzyme was made, that will help you to remember which pH it's supposed to work well at. Hopefully these 2 videos have helped with some studying you're doing, or if not, maybe you can let me know how I could improve them.